Okay, so we have seen this, the sympathetic nervous system. Where is it? The sympathetic nervous system from the okay. Does anybody remember from what level of the spinal cord or vertebral column do you get the preganglionic fibers which are coming out and going to the heart? T1, T2, T3 and T4, that is it. So, from here, thoracic 1, 2, 3, 4, okay, the spinal nerves which emerge from that area go to the preganglionic, the preganglic, and then the postganglionic fibers will release nor-epinephrine on the heart, and we have done that. Okay. So, this is a block diagram that tells you how the changes will happen at every stage, and so you can so just see baroreceptor reflex. If the pressure falls, let us follow this through. Baroreceptors, pressure falls, stretch on carotid sinus, downward arrow means what? Reduce, then what? Firing rate for carotid sinus will go down, as we have seen in that slide. Then as a result of that, parasympathetic activity will also of the heart will go down. Okay, are you with me? This will go down, therefore the heart rate will go up. Means the okay. So you are sitting in the medulla oblongata, okay, and you get the information that the pressure is not good enough. So the first thing that you do is, okay, shut off the parasympathetic nervous system. So is the heart getting enough acetylcholine now? No, it is not getting enough acetylcholine, okay, so it is not inhibited. In this particular case, acetylcholine is acting by what receptors? Silence. Acetylcholine, muscarinic, M2, what is it? Yeah. You know something, what we have seen so far are rapid changes. Rapid may be immediate in seconds or in minutes or in hours. Okay, we have seen. There are some of the changes which happen over a very long period of time. Okay, and the classic example or the classic experiments that are done is, you know what they do? Very elegant experiment. You take the fertilized chick eggs. Are you with me? Fertilized. So is the development happening there? Yeah, development is happening there. Okay, then what you do? You take 2 incubators. Are you with me so far? In this incubator you have normal air. In this incubator you have air which has less of oxygen. So if this is normal and if I presume this is 20% oxygen, am I okay there? Hello? Roughly give and take something. 20% is here. I'll in this chamber, incubation chamber, I'll have 10% oxygen, and then I'll put the eggs in both. Okay, and then I'll let it develop for say 10, 15, or don't go to the end. You see, just for first 10 days, and then take out both the eggs. Okay, and then I open both the eggs and put them under microscope, or even without putting them under microscope, I find that in this egg, which was incubated in what? 10%, I find that there is far more, the number of blood vessels is very high here as compared to this. What has happened? Can somebody guess? Just the number of arteries, veins, capillaries, everything is much more. Means the blood vascular system is sensitive to the amount of oxygen, and when the amount of oxygen is less, our physiological systems compensate by giving what? By giving what? Increased your own RBC count may be 5 million, okay, per cubic mm or whatever, may be huge. If you go and stay at higher altitude, like in some place in Himalaya or Lake, over a period of 15 days, you will find that your RBC count has gone up. Why is it going up? Why has it gone up? Because when you go there, your physiological systems realize that there is less oxygen, so you try to compensate. It takes time, okay? It takes time, but you are able to. And the last is actually amazing. We never think of these problems, you know, but they are there. The problems are there. The problem is, there is a case of birth of a baby. Are you with me? And the baby is not doing too well, so you put the baby in incubator, okay? And you want to make sure that the baby is well protected, the baby is not able to respire properly on its own. Therefore, you increase the supply of oxygen. So, where is the baby? Actually, I will say that the baby is in oxygen tent. So, the baby is getting plenty of oxygen, no problem. So, whatever its development is happening. Now, after, you can't keep the baby there forever. After 15 days or 1 month, you decide to take out the baby. The moment you take out the baby, baby experiences that oxygen is far less. and then there is a profound rebound in the circulatory system, and there is so much of blood vessels that some of the blood vessels, the number of blood vessels increase elsewhere, it does not matter, but they may increase in the retina, and if there is increase in the retina, then it may those blood vessels may obstruct the vision. Hello, are you getting the argument? Are you getting the argument? Okay. So this is a very, so you can talk about the changes over a period of longer period of time. Very interesting point. Read about it. Yes, please.
The change in development caused in the chick's case, does that reverse after like hatching in a normal environment?
It does. It goes down. It does go down. Yeah, it is. I mean, those changes are happening because there is a certain trigger. Yeah. Okay. When you remove the trigger, eventually the things will come back to normal. But I don't know how far. I mean, how far if you have gone, it will come back. That I don't really know. But you have to work. But literature is there on that issue. Okay. Now I will talk about a very interesting issue, and I want to. We always talk about lymph, lymph. Okay. Let's talk about lymph. It is one of those topics which we know is there, but we generally don't talk about it. Am I right there? Always in a book, there is a topic, I know it's there, I know somebody says it's important. I also did it, I also do it, I mean, see, not that, okay. But then when we do that, okay, in our primary instinctive behavior towards the studies, okay, we miss fun, okay. So therefore, I am going to talk about this lymphatic system. You know what happens? If I ask you, tell me the name of the organ where ultrafiltration is happening, you will instantly tell me kidney, okay. Okay. Am I okay there? Can you tell me the name of the organ where ultrafiltration takes place? Kidney. I am going to repeat my question. Can you tell me the name of the organ where ultrafiltration takes place? Kidney. I am not getting the chorus, You see that I'll tell you why. That chorus gives me a kick, and you are depriving me of that. So tell me the name of the organ. Okay. You know something? At the level of every capillary, small amount of ultrafiltration is happening. Very interesting. Every capillary, small amount of ultrafiltration is happening, and the fluid flows out. And why do the fluids flow out? Because there are capillaries, and there are capillary endothelial cells, and some of the endothelial cells have gaps, okay, and through the gaps the fluid can go. Through the fluid, okay, here is a capillary. Okay, and in the capillary there is an endothelial cell, there is another endothelial cell, and whereas the oxygen and oxygen can safely go out, and whatever, and carbon dioxide can get in, and nitric oxide can get in. There are small molecules like amino acids and other molecules, glucose, etc. They can just go through this gap. Okay, and there are some molecules which have carrier in the endothelium, so there is lot of trafficking can happen, etc., etc. But the point is, the small molecules can go in and out. But large protein molecules cannot go, we have seen that already. When we studied capillary, large protein, so large protein molecules are confined here, okay. But is the system so perfect that no large molecule will go? No system is perfect, okay. So there is some loss of large molecules also. So you can have ultrafiltration here. Why ultrafiltration? Because just as the blood is about to enter into the capillary, the blood pressure is about 30 mmHg. And given the fact that capillary endothelial cells are extremely thin and delicate, and the spaces, the fluid is going to come out, okay. And along with that fluid, some large amount of proteins are going to come out. Now, what will happen if you allow the proteins to stay there? Bad idea. You cannot allow the proteins to stay there. Therefore, and all the substances which have come out as a result of ultrafiltration, the nature has provided us with an amazing system wherein those substances, there is another system of tubes, another system of capillaries and blood vessels which return those substances back to the circulatory system, and that I can show you by way of, in this image you can see how the system, how the substances can flow out. Okay, so here we have the capillary. Where is this capillary network? It can be anywhere, muscle, pancreas, anywhere. and here is the oxygenated blood comes here, goes into capillaries, and there is a system of, there is certain fluid that is flowing out of the capillaries under the influence of higher pressure, and that it gets into tiny tubes, and then these tubes, it flows through the tubes, okay, and finally it is poured into the vena cava, superior inferior vena cava, and then finally it is returned to the blood. Okay. In this you will never get an RBC, okay, because RBCs are very large, but you get plenty of WBCs. You get you get large protein molecules, and you get a lot of filtrate. Now remember this. I will take you back to this. Is interesting. Focus on this. We are look at the bottom. Can you see the capillary there? Okay. What do you think is the distance from this end to that end of the capillary? How much? How much? 1 millimeter. Okay. All right. And if I measure the pressure here, okay, it's going to be how much? 30. And how much? What is going to be the pressure here? Or that is actually represented by this green line. So these 2 points are represented by the. Can you see the green line there? And can you see, go to that y axis? So what is the pressure of the blood as the blood is entering into the capillary? Read there.
30.
Little more than 30, 32, 33, whatever that author is showing. And by the time it goes to the other end, where are we? We are here. We are about, say, 12, 13, 14. Are you okay so far? Now, as a result of the fall in the blood pressure, rise high and then going down, the material is getting filtered out of the capillary, ultrafiltration. And how much of blood is being filtered out can be judged by way of the length of the arrow. Now, why can you guess why this arrow is slightly longer than the second arrow? Higher pressure, lower pressure. Okay. And in the third, in the pressure is almost is less. Okay. Now, just as now get the point. Follow this. Just as the pressure, hydrostatic pressure, hydraulic pressure, okay, generated by the heart is going from 32 or whatever to 14, whatever is going down, okay, and as a result of ultrafiltration, okay, some of the protein molecules are going, few of them, but they are also going, okay. Now let us look at the blood. The blood, as it is available at this point, has a certain osmolality. Are you with me? Has a certain what? Osmolality. Has a certain osmolality. As the blood flows, okay, a fraction of a millimeter by fraction of a millimeter by fraction of a millimeter, what is the most, the watery part will flow out easily. Large protein, few, but not many. What is the dominant factor that is flowing out? Watery part. Large protein, 99% of the large proteins are still confined to that. So by the time you reach here or here, what is the physical composition of the blood? Less water and more large proteins, which means more osmolality. Are you with me? Now, so as I go from here to here, the osmolality is increasing and the blood pressure is decreasing. Are you with me? So I am getting exactly opposite picture from the first half of the capillary versus the second half of the capillary. More blood pressure, less osmotic pressure, more osmotic pressure, less blood pressure. As a result of that, a large amount of fluids which are going out here are getting in there. Hello. Just imagine how much of drama is happening within 1 mm of the length, and given the fact that RBC takes less than a second to go. Okay, but within that time, this continuously the fluid is coming out and continuously the fluid is going back. Okay. This was described, this we call as what? Starling hypothesis. What do we call it as? Starling hypothesis. Now, this Starling name is coming for the second time. When did it come for the first time? Say that loudly. Frank Starling hypothesis, or Frank Starling mechanism of heart or law of heart. Have you forgotten that mechanism? Lost, watered down, gone for good ever, goodbye. Remember that, huh? Okay. Same Starling, Ernst Starling, a British scientist, and the time is about 110 years from now. 1910 or something like that. Okay, he discussed the he discussed the phenomena. He described the phenomena that across a capillary, the things come out, come out, come out. They go in, go in, go in. Okay, and it has been found that over a period of 24 hours within a human body, the total amount of filtrate that comes out of all capillaries is about 20 liters. It's a huge quantity. Out of that 20 liters, about 17-18 liters goes back. Comes out in the earlier part of the capillary, goes back in the latter part of the capillary. Out of 20 liters, 18 liters has gone back. How much has remained? 2 liters. 2 liters has remained. If you don't return the 2 liters to the blood vascular system, you will have swelling in the tissue, edema in the tissue. So as to make sure that you return that 2 liters over a period of 24 hours back to the circulatory system, you have that lymphatic system which I have told you. Are you okay so far? Hello? You have got the philosophy of the lymphatic system. Okay, lymph is generated at every capillary at every moment. Certain microliters is generated. Okay, and lot of it goes back. 80% goes back. 80-90% goes back. But what doesn't go back? Okay, for that we have the lymphatic system, and that lymphatic system is not just that. Eventually, lymphatic system has lymphatic nodes, and those lymphatic nodes play a very important role in what? Immune system. Okay, so that is a part of the immune system. This is approximate numbers, you know, what we have seen in the previous slide. The author is trying to give us some numbers. What are the numbers? The blood pressure in the earlier part of the capillary is about how much? 32. Is the effective pressure 32? Not really, because outside also there is fluid. In that outside fluid also there are salts, and there is also some protein that is also generating certain osmotic pressure, and that pressure is about how much? About 22. So what is this minus this will be how much? So the fluid is going out 10. It will actually, this is 10 is absolute. It will be reducing, you know, from capillary to, as you go to the center, it will be reducing, and as you go from here, okay, the blood pressure has gone down. Okay, this is more or less remains 22 or whatever, and then as a result of that, the net pressure inside is -7. So the fluid will go back. 100%? No. 80 to 90%. Whatever 10-20% still remains, it has to go where? It has to go into the lymphatic system. And I wanted to finish, but it seems it's difficult for me. But in the next lecture, I will try to finish the lymphatic system. And